Hey everybody. Today we're looking at section 8.2, which is cell structure. And if you remember what we did in section 8.1, this is an example of something called a eukaryotic cell, which is the same type of cell that we contain throughout our entire body, right? Eukaryotic cells, remember, make up the majority of those kingdoms of life that you think of when you talk about living things. All animals, plants, fungi, and protists are made up of eukaryotic cells. In contrast to that, prokaryotes, remember, are strictly bacteria. That would be your only example. So when we look at cell structure, the majority of our conversation today is going to be involving specifically eukaryotic cells and the components found within them. Okay, so go ahead and get the heading. And then looking at here, our learning objectives, you can see this is going to be a fairly long section today. So we're looking at the function of different organelles in terms of the nucleus and things that are designed to build proteins and then move proteins around. And then the functions of other types of organelles like the energy producers, chloroplasts and mitochondria. And then the additional ones like vacuoles, lysosomes, cytoplasm, and so on. Cell membrane is going to be a big piece of this because the cell membrane, though it's not really truly considered an organelle, it is a very important structure in a cell. And so that's where we're going to kind of finish today. And that'll lead us into section 8.3, which is coming up in a few days. Okay. So exploring cell organelles. So you definitely want to get all of the information here on this slide, the characteristics that all cells share in common. And this is for everything. That includes both prokaryotes and eukaryotes. All cells have a cell membrane. All cells, by definition, have cytoplasm. All cells, by definition, also have to have ribosomes. So I'm going to put up here, as a reminder, this is both prokaryotic and eukaryotic. So anytime you hear any of these structures or organelles mentioned, recognize that that is true for every single living thing in every single cell that we find. So the job of the cell membrane, I mentioned we're going to come back to towards the end of this section. The cytoplasm, it's a jelly-like substance. You've maybe heard that song where it talks about cells are made of organelles. One of the things that they say right after that is that the cytoplasm gels, because it's like a gel type material, and that helps to suspend all the organelles and reactants and molecules that we find in the interior of a cell. So really almost everything interacts with the cytoplasm. All cells also have ribosomes. We're going to talk about their function here in a second, but those are basically these little tiny dots that you see over here kind of floating around the cytoplasm. And they have one really important role that would be consistently used across the board: animals, plants, fungi, protists, and even bacteria. So all of these are found in both prokaryotes and eukaryotes, okay? So at the bottom here, you see a plant and animal cell. Some of the organelles we're going to talk about will be specific and only found in one and not the other, but you can see kind of the coloration and all the different things that we find here. A lot of it's going to be very, very similar. So we know that all eukaryotes have organelles. Notice here the picture didn't change at the bottom, but some of this text did. So you're going to want to make sure you get those items there. Organelles are tiny structures, tiny spaces really where specific jobs can be done. So it's a way by which a cell can kind of organize what needs to get done in order for that cell to be able to survive. So we see lots of different things in these images, like I mentioned before. Some of these, again, are membrane bound, or I should say the majority are. But they all kind of share the same responsibility, which is that they have one primary function, and their job is to carry out that one function. So cells themselves can make more organelles, and they can destroy organelles when they're no longer functioning properly. And different cell types have different amounts of some of these things. So you can see here one of the major differences between this plant and animal is plant cell has this really large kind of bubble-looking organelle, and animals in comparison is about that big. Those are called vacuoles, and those vacuoles we'll come back to here in a little bit and talk about what they do. But in plants, its main job is to hold a lot of water, and in animal cells, we just don't need to be able to do that. So that's why ours are reduced in size. In general, these organelles add to the complexity of eukaryotes, making the processes that we do in our cells much more efficient than what we see happening in prokaryotes, which are just kind of doing everything out in the cytoplasm together. So there's a real benefit in this case to being eukaryotic in that it helps to kind of further control what's happening and also organize it in a meaningful way. Okay. So if we go on and actually start looking at specific organelles, the one we tend to always start with is the nucleus. So the nucleus is going to be kind of like the control center of the cell. That's usually what I hear when I ask students what the nucleus does. So certainly you're going to want to get both of these bullets here. But in reality, what the nucleus does is not to strictly control the cell, but its major job is to hold DNA. And then within that structure where it's holding DNA, it helps to provide a place where enzymes can come in and start to read the DNA code to create things that we call proteins, which is something we talked about in chapter 2. So the nucleus is always our starting location. It's where we're always going to begin what we call protein synthesis or making proteins. All right, so the first few organelles we're going to talk about, including the nucleus here, are going to be involved in the process of what we call protein synthesis or the creation or making of proteins. So you can see here my plant and animal cell in these images here on the right. Both of them have a pretty well-defined nucleus, and that nucleus is something that if you look at a cell under a microscope, it's something that usually is going to stand out, okay? In terms of the nuclear structure, we have little pores. Obviously, this is a membrane-bound organelle, meaning it has its own little membrane around it, and you can see here how it's kind of cut away. The nucleolus here is where we actually hold our DNA. Okay, and then we have this envelope or this membrane that surrounds it as an additional layer of protection. Chromatin, we'll talk about a little bit more, but chromatin is DNA condensed with histones, and that'll become more meaningful when we get to that a little bit later in February and March. Okay, so moving on. So nucleus is where we're going to start. We read the code here to start creating proteins, and once that happens, and you can see here we're representing again my nucleus here on this side, and let's say I have DNA in here. That code gets read in the form of something called mRNA, and then that mRNA is going to actually leave the nucleus and go out and visit a ribosome, okay? So ribosomes play a really important role in making proteins, and this is the reason why we have these in all cells of all living things, because all living things need proteins. DNA is how you inherit information, but protein is how you show or express that information. So to actually develop a function in a cell or be able to survive in terms of a living thing, you have to express the right set of proteins at the right time. And so ribosomes are helpful in that way, because without them, we would not get proteins being made in the first place. So proteins get assembled on ribosomes, and you should definitely know that is the role of ribosomes, is to build proteins. All right, now as we go through this process, some ribosomes can be freely floating, and some ribosomes are attached to something that we call the rough endoplasmic reticulum, or a lot of times we often refer to this as the rough ER. Okay, so there's a smooth ER and there's a rough ER, and I believe I'm going to come back to smooth ER in just a second, but let me check. We'll look at it in a second. So ribosomes can either be freely floating or they can be attached to this rough ER, and you can see my rough ER is this little kind of folded membrane here surrounding my nucleus. Okay, so the rough ER holds some ribosomes like we see right here. That DNA or mRNA can be pushed in that direction. The rough ER helps to build proteins also. So usually what I say for a definition for the rough ER is that it works with its ribosomes to help build proteins. And then beyond that, if we want to take proteins and actually push them out of the cell or use them within the cell for something, they're going to route themselves through what we call the Golgi apparatus. And the Golgi apparatus, its job is to sort, modify, package, and kind of label all of our newly made proteins. So sorts and packages proteins. So that's the job of the Golgi. I usually like to think of the Golgi as being kind of like FedEx or UPS or the US Postal Service, where you could actually take something that needs to be sent somewhere, you bring it there, they're going to package it up, and then they send it where it needs to go. That's very similar to what happens in a cell involving a protein. So when we go on and go to make a protein that's going to have a specific job, the cell's going to kind of direct it along the pathway that it needs to go. So we could say we start in the nucleus, we go from the nucleus to the rough ER and the ribosome. The ribosome actually creates the protein and reads the code. And then from there, we take that newly created protein right here in the Golgi, and we're going to start to push it out of the cell or push it to a different location in the cell using the help of this last organelle here, a vesicle. So vesicles, for me, what I always like to think of, these are like vehicles. Their job is to transport things throughout the cell or to the cell membrane and then beyond. But for me, when I think of vesicle, that's a word that comes to mind, is vehicle. So if that's helpful to you, then certainly do that. But if we're equating to the Golgi, or if we're assuming that the Golgi is like UPS, and the vesicles would be like the brown UPS trucks that carry packages to different places. Okay. So there was a whole bunch of organelles on that. I would definitely make sure you've got that information. And I have some like additional reminders here. So let me go back to this one. So here's our Golgi apparatus. On this side of the image over here, you would have the stuff that we just saw back there. So we had the rough ER and the ribosomes, and then we had our nucleus, right? And all those push the protein here. So here's my Golgi apparatus in blue. Again, it looks like a set of folded membranes, but it's not rough anymore because we don't have ribosomes attached to it. These are just smooth folded membranes. And there's where we're actually going to modify, package, sort, put a label, a tracking label, if you will, on our protein. And then we go on to get it shipped and sent somewhere with the help of vesicles. So here is actually a vesicle. They're basically little empty compartments where we can stick proteins or anything else that needs to be transported from one place to the other. They are membrane bound, as is the Golgi, as is the rough ER. So that means, again, they're only found in eukaryotes. But that membrane kind of protects what's inside, and it helps to move things from one place to the other. Okay. So those are kind of the organelles that generally are involved in the production of pathway, or the production of proteins. This is the pathway that we use. So here is that general sequence of events. So we start again in the nucleus with our DNA. That would be step one. That DNA is made into a copy, which we call mRNA, and that goes out to a ribosome, either one that's freely floating like this, or is attached to the rough ER like here. So this is my step two. In any case, either way, those ribosomes, whether they're working with rough ER or not, the job is still the same. They're still going to build a protein. Ribosomes that are attached to the rough ER, obviously they work together in doing that. From there, we go on to the Golgi apparatus. All right, so the Golgi here is my third step. And notice I'm using vesicles to get from my ER to my Golgi. And then we end up with vesicles here again at the end in step 4 that are going to push our proteins out of our cell. Now, I left off a couple of things here. Let me shift this up here. So one of the items that I forgot to mention, or that I did not mention yet, was a smooth ER. And the reason why it wasn't brought up yet is because the smooth ER's job is very different. Even though it's still the endoplasmic reticulum, it's still an ER. The job of the smooth ER is not like the job of the rough ER. So the smooth ER, notice what it looks like here compared to what we saw in the rough ER. There's no bumps or dots. That's why we call one smooth and one rough. But with the smooth ER, its job is completely different. It's also responsible for making things, but it makes lipids. So it's going to make triglycerides. It's going to make phospholipids. It can even help to make cholesterol and things like steroids from those cholesterol precursors. Smooth ER also has another role in that it also helps with something called detoxification. So if you know anything about detoxification, that really involves the removal of things that could harm the body. So organs in our bodies, like the liver, have cells that are just full of smooth ER because their primary job as being part of the liver is to remove things that could be potentially harmful to the body. And so the smooth ER helps with that. All right, so the smoothie R's jobs, again, make lipids and do detoxification. It's not involved in protein synthesis or creation at all. Okay, so definitely make sure you get that part jotted down because I don't think I am coming back to that in this PowerPoint. Okay. All right, so switching gears a little bit, if we go on to other types of organelles, we know that we have organelles in all living things that play a role in either creating energy or converting energy that's been consumed by a heterotroph. So the first one here on our list is a chloroplast, and the chloroplast, I'm sure, is something that you've heard of before. Remember, chloro was a Latin root, and chloro means pale green. So if that's helpful to you, jot that down. It is pale green because that is truly the color that this organelle is. The light that's being reflected is actually green. And we'll come back and talk about that more as we get into photosynthesis in chapter 9. But the job of a chloroplast is to capture light energy and convert it into food or stored energy in the form of things like glucose. So remembering back to what we did in chapter 2, glucose is a monosaccharide. It's super important in all living things, regardless of if we're talking about plants or not. Even when we consume something, glucose is what we transfer from our food to be used in our cells for actually usable energy, okay, what we're going to call ATP. And we'll come back to that again in chapter 9 and also in chapter 10. So the process by which chloroplasts do this is called photosynthesis. So usually if I ask someone to identify what's the job of a chloroplast, they tend to go with to do photosynthesis, okay? So here's what they look like. These are specific and found in plants only, all right? And this is truly plants and photosynthetic organisms, right? Anything that's photosynthetic would be certainly containing chloroplast. But within eukaryotic cells, and this is one of those, you can see this is definitely membrane bound, right? Look at this membrane structure here that's been kind of cut away. Because it's membrane bound, it's eukaryotic specifically, but again, it's only plants within those categories. Okay, so this is one that kind of stands out. We do not have chloroplasts in our bodies. We cannot do photosynthesis, right? What we do have, though, are mitochondria, and you can see here from the image on the right, one of the things that typically kids tend to forget about, you have your animal cell here and your plant cell here, and when we look at these, you want to keep in mind that plants not only have chloroplasts, they also contain mitochondria. So mitochondria, their role is to take chemical energy in the form of glucose and convert it into energy molecules that the cell can actually use. And those energy molecules are what we call ATP. And we'll talk about that again in the coming chapters. But ATP is actually the product of this whole process. And the whole process that it's doing is called cellular respiration. So all eukaryotes can do cellular respiration, all plants, all animals, all fungi, all protists. Okay, these are going to be important processes when we look at those next few chapters that are coming up. Okay. So that's a mitochondrion. When we switch gears a little bit here again, here are some other organelles that have another variety of functions, and they're not related necessarily to either energy production or to protein production. So these are slightly different. We did talk about vesicles already, and they're reappearing here on this slide because, again, they can transport lots of different molecules, not just proteins. Okay. So vacuoles, and by the way, on this slide, I would definitely, get all of this. Vacuoles are fluid-filled organelles. They store materials. Plants usually have really big, large vacuoles, like I showed you on one of those first slides here of this section, and that's because they need to be able to stand upright, hold a lot of water, and maintain that water storage. And so those vacuoles that they have really kind of swell up and put pressure sometimes on the cell wall, which is what allows a plant in its kind of green stem to stand right up. It's not like in a tree. We're talking about like grasses and dandelions and that kind of stuff. The ability of those things to stand up is based on their ability to fill up their vacuoles in their cells. Vesicles, like I said, can transport lots of different things. And we have lysosomes, which is kind of a totally different function. But lysosomes play a role in getting rid of materials that a cell no longer needs. All right. So if we think about, again, another Latin root here, lyse. Lyse meant to split, loosen, or break, right? So split, loosen, break. So the idea with lysosomes is that they will break apart molecules, they will break apart organelles, they will break apart infectious cells like bacteria or viruses and get rid of them before they could potentially harm what's going on in the cell. Okay, so lysosomes are helpful in that way. They help to break down things that are no longer needed. And we usually think of these as kind of like a garbage disposal of a cell, right? So the thing in your sink, you flip it on, it kind of chops up all the food and it pushes it out from your sink in your house in the plumbing. Same idea with lysosomes, right? They're going to chop everything up. They split, loosen, and break molecules apart, and then they send them on and out of the cell with the help of their vesicles. Okay, so lysosomes are unique in that way. All right, I've got a couple more organelles, and then we're going to move on and talk about the cell membrane next. Okay, so cells have an internal structure. This is provided and maintained by something that's called a cytoskeleton, and it's what you think of it would, possibly be, which is that it provides some support, it provides some shape, it allows, in this case, the movement of different molecules and vesicles along tracks of these tiny little fibers. So cytoskeleton works with these things called microtubules, which are tiny tubes, and microfilaments, which are tiny filaments, even smaller than microtubules. And they will perform lots of different functions. What I usually say when we get to this slide is I have students write down like 2 of these functions. And that would be fine if you want to go that route, or of course, if you want to write more, that would be fine too. But the general idea is this is like a skeleton for us that's in the cytoplasm of a cell. So it helps to support and shape, again, position and move organelles with the help of things like vesicles, and will assist with cell division. We'll talk about this in chapter 11 when we get to about winter break. Okay, so mitosis and cell division are going to use cytoskeleton pieces, like little microtubules, to help with that process. It can also help in cell movement with additional kind of extracellular organelles like cilia and flagella, and we'll see those here on the next slide. Okay, so this is, again, cytoskeleton. This is, again, specific to eukaryotes, and it has these specific functions. But again, it's on all eukaryotes, not just plants, not just animals, but all eukaryotes. So when I mentioned the fact that this cytoskeleton plays a role in cell division, this is specifically what I was referring to. So centrioles and centrosomes, these things actually interact with the cytoskeleton. They kind of make up part of the cytoskeleton because their components, the microtubules that make up these centrioles and centrosomes, are actually part of the cytoskeleton. So the job of these is to help with mitosis, and they also create and kind of combine to form these cilia and flagella. So down here at the bottom of the screen, this is cilia on the left, and then this is flagella on the right. So these are things that sit on the outside of a cell that project outside the cell wall or the cell membrane. Their job is to help these organisms that are maybe single-celled to maneuver through their environment. So they help with whole cell movement, or they help with the movement of things around a cell. So cilia, as you can see right here, are kind of hair-like in their structure. There's a whole bunch of them. Okay, on the outside, it's all the green kind of thread-like stuff you can see there. It kind of makes it look fuzzy. Flagella help with the movement of the whole cell. This is the type of structure that we see on sperm that helps sperm cells to move when they're trying to go and fertilize the egg. Most cells have 1 or 2 flagella that require these. They don't usually have 4. This guy's kind of unique here. But these we see all the time in protist-type organisms, things that are single-celled and that have a need in their environments to allow them to be able to move, right? When we look at our bodies, I mentioned sperm have flagella, but we also have some cells that have cilia, and those are the ones right here that line your trachea. And so what those guys are doing is they're constantly moving. These are little tiny hairs, like I said. They're constantly pushing like this in your trachea, which is the tube that connects your mouth to your stomach. sorry, not your trachea, your esophagus, the tube that connects the mouth to the stomach. And they're constantly kind of beating like this. And when they do that, they're pushing saliva down, they're pushing mucus down, they're pushing food down, and they're preventing it, hopefully, from coming back up. Now, obviously, sometimes it does come back up, right? But that doesn't mean your cilia aren't functioning. It's just the normal kind of movement for them is pushing things downward, and that helps to get everything down into the stomach. So that's kind of cool. Centrioles and lysosomes are only found in animals. All right, just like we saw, chloroplasts are only found in plants. So these are both eukaryotic specifically, but they are only, in this case, for centrioles and lysosomes, only found in animals. Now, there's one additional organelle that's found specifically in plants and not in animals, and that is our last organelle here for the day. Not the last slide. We still got to talk about the membrane, but our last true organelle. So the cell walls that we're looking at here, and this is an image of what a cell wall looks like. These are found in some eukaryotes, plants and fungi have them, some protists would have them, and almost all prokaryotes. So you think about something that's single-celled, like a bacterial cell, or a yeast, for example, or an algae. When we think about those types of cells, they better have a cell wall because that provides some additional protection. So if your whole being is contained within one teeny tiny microscopic cell, you want to be able to protect that. And this is one way that they can do that. So in some eukaryotes, plants and fungi, again, most prokaryotes, they provide rigid structural support. Cell walls, remember from our content in section 2.3, we talked about one polysaccharide that we find in cell walls, which is called cellulose. And cellulose is made up of a whole bunch of glucose linked together, but it's really kind of rigid and allows these things to kind of stand upright, as we see in things like plants. And you can see here's a kind of a cross section of what we see in a cell wall, and you can see all the different components that make that up to kind of provide that rigidity. But these are also, again, not found in animals, but strictly are in those that need that additional structural support. We don't typically need that because we're not relying upon the same things that really plants and fungi and protists are. Okay. So those are kind of all of our organelles. The last thing we have to talk about in this section is this final cell structure, which is a cell membrane. So the cell membrane has two primary functions. It's super important. Okay, when we look at any cell structure, this is one of those defining features that all cells, again, share in common, but is really, really important in that way. So the major functions for a cell membrane is to form a boundary between the inside and the outside of the cell, and to also control the movement of things into and out of the cell. So what we're looking at in this image here is actually a cell membrane that we have kind of cut away, right? This is like as if you had a cell here that's spherical, and you cut it in half this way, and then you're peering into one of the cell membranes on one of the edges. Okay, so we're looking down into that cell membrane. So you would have more cell going this way, you would have more cell going this way. This would be the inside of my cell, and then this would be the outside of my cell. All right, and then within it, we have all sorts of different molecules. So we have these things here called phospholipids, which look like this. They kind of take on this structure that resembles something like that. Those phospholipids obviously are a type of phospholipid. They were one of the examples of a phospholipid that we talked about in chapter 2 as a polymer. They have two fatty acid tails here, right? And then they have this one phosphate head group here. We also have some membrane proteins. Those are these. This is called a transport protein. We have some cholesterol that's going to stick out here. These little yellowish golden kind of colors. These are cholesterol molecules. We have another kind of embedded protein that can be maybe a receptor. So we could call this a protein receptor. And then we have, again, here's some more of that cholesterol here. And again, we can have some carbohydrates in some places as well. So in reality, we've got carbohydrates involved here. We have lipids involved. We have proteins involved. The only biomolecule that we've talked about that's not part of the cell membrane is going to be a nucleic acid. And we know that they're not found here because if you remember, they're always found in the nucleus. Okay, but everything else plays a part in kind of creating the structure and the function of this membrane. So if we look at how this actually is structured, let me go back to this a little bit. So we have 2 layers of phospholipids. So if you notice what we're looking at here, my phospholipids, I have one series here where they're what we call their head groups here, their spheres are facing up, and then I've got another one where they're kind of facing down. And what that means is that we have what we've created as like a bilayer. There's one where it's kind of facing, the head groups are all up here. The other one, they're flipped kind of upside down, so the head groups are all down here. What we know this means is that inside this membrane right here, we have our fatty acid tails. And if you remember anything about our fatty acid tails, these things are made up of lots of carbon and hydrogens and no charges. All right, and so what that means is that these things are not polar. These are non-polar. All right, and we know that non-polar things like oil, like grease, do not like to interact with water. And so these little fatty acid tails, being that they're non-polar, they're not interested in being around water. And so the way that these membranes kind of spontaneously will form is so they're not interacting with water. So on the outside of my cell, remember we have lots of water in our bodies. On the inside of our cell, we also have lots of water. But within this actual membrane, there is no H2O. So those fatty acids that are non-polar are kind of kept away from water, which is exactly where they want to be. These little guys, the little pink spheres here on the top, these guys are called phosphate head groups, and they're kind of unique in that they have charges. So these guys are phosphate. Well, that's not going to work, is it? These guys here are called phosphate head groups. Let me erase that and rewrite. Maybe if I can get it to right here. There we go. So not writing. Oh, let me fix that. Sorry, guys. Okay, so these guys right here are called phosphate head groups. And they're called this because they kind of look like heads on these little stick figures. But those phosphate head groups themselves have this as their formula. It's PO4, so 4 oxygens and 1 phosphate, and a tiny negative charge. Now we know that for things that are charged, they like to interact with water, right? These phosphate head groups, and I'm going to go ahead and kind of color purple here so we can keep them straight. They actually have charges, which makes them polar and which makes them happy to interact with water. And so they orient themselves in this membrane, again, facing the water on the outside and facing the water on the interior of the cell. Okay, there are 2 additional words that we use to kind of represent these different, the kind of directionality of these phospholipid molecules. And those are to say that these polar head groups are actually hydrophilic. And this is incorporating some of those Latin roots that we've learned before. Hydro meaning water, and phil meaning to love something, okay? So hydrophilic polar phosphate head groups face the outside and the inside of the cell so that they can interact with water. On the other hand, fatty acids, because they don't like water, because they're nonpolar and they lack charges, we call these guys hydrophobic, which means they have a phobia of water. They're actually afraid of being around water, and that's exactly why they flip, so that they've got their fatty acid tails kind of facing the interior of this membrane. Okay, all of this is really important because this determines how things get into and out of a cell. The properties of this membrane determine what can move and how they're going to move. Okay, so understanding this is going to be really important. And if I were you, I would draw something that kind of illustrates what I'm representing here. Okay, so at least something where you're labeling what's a phosphate head group, where are the fatty acid tails, and how does that relate to the water that we see that makes up all living things. Okay, so kind of adding to this, as we look at those other molecules that I mentioned are also found in the membrane, we want to identify what their functions are. So the phospholipids that we saw, and here they are down here. Again, we've got our fatty acid tails on the inside and our phosphate head groups on the outside. But all these other things that we see kind of embedded and interacting with this membrane, they're all there for a specific purpose. So the phospholipids kind of help to kind of orient everything, and they help to provide some just general structure and eliminate things from entering that maybe shouldn't enter. But there are others that do other things. So carbohydrates we can see right here, and I'll use a different color for this. So carbohydrates right here, these guys have a unique function in that they help with cell recognition. And what I mean by that is if this is like if we're saying that a cell is equivalent to maybe a human, this is like a name tag or an ID badge. Okay, so if you think about two cells, if they're floating through an individual, floating through a living thing, or in two different locations of a living thing, these name tags will help them identify who's who, because cells can't see into other cells, right? Obviously. So it's kind of like a house number on your house, maybe, that identifies what address your home is actually at. But it is strictly like a name tag. Proteins can play a number of different roles, and let me just go ahead and label this. So we'll do blue carbohydrates. Proteins can do lots of different things in cells, and when we talk about in a cell membrane, their major job is to either function as a receptor or a transporter. All right, so receptor or transporter, and later on in chapter 8, you'll see what we mean by receptor, excuse me. Transport we're going to look at right here. So these guys down here, this is a transport protein. Its job would be to either take things into the cell this way or push them out that way, or potentially could do both. Either way, these transport proteins allow things through the membrane that cannot cross on their own. And we'll talk about what those mean and what those properties would be on the next slide. Same here. Here's another transport protein. Notice it's got a different structure. But that would have this similar function. This guy here, on the other hand, these look like they're probably membrane receptors. Okay, and again, I said we'll talk about what those are in a few days when we get into 8.4. Okay, but their jobs are to either absorb a signal or to cause a change in something occurring on the outside of the cell. The last thing we see here are our cholesterol molecules. And cholesterol molecules are not really even being shown in this image, but in reality, they would be kind of scattered throughout our phospholipids. And so I'm just putting some little red lines. Remember, cholesterol is an example of another type of lipid. Its job in this case is to help maintain membrane fluidity. So what I mean by that is these membranes are not just kind of stagnant and sitting around. These membrane pieces are all kind of floating in the same space together, and they float and they bump into each other and then they move down. And so what that means is that this cholesterol helps to maintain that level of fluidity and movement, even if temperatures kind of drop, because cholesterol is similar to something like an unsaturated fatty acid that can maintain and remain liquid in different temperatures. Okay, so this is helpful in a way to allow things to kind of shift and move in response to something happening in the body. All right, so we have carbs, proteins, cholesterol, again, everything in these membranes except for nucleic acids. And overall, those highlights. Okay, last slide of section 8.2. So the membrane, like I said, one of its major jobs is to prevent things from entering that shouldn't be in, and also push things out that need to get out. So we like to say that a membrane is considered selectively permeable. And what that means is that only some molecules can cross the membrane on their own. It's selective in terms of what can permeate or cross the membrane. Okay, that's where those words kind of come from here. We know that when we talk about cell membranes, in general, the smaller the molecule, the more likely it is that they can cross the membrane on their own. These two lists here are going to be super important for you to jot down, and certainly you want to make sure you also get the definition up here. But in terms of what can and cannot cross, this is something that if you're in my classroom, I ask you a number of times as part of this chapter leading up to your test, okay? So we know that things that are smaller can cross. We know that if you're hydrophobic, that you can cross. And remember, hydrophobic things, like we just talked about with the phospholipids, this would be like the fatty acid tails. These are uncharged and nonpolar molecules. So that means anything that can cross has to be able to interact with this, my nonpolar, uncharged fatty acid tail interior of my membrane. Excuse me. And the reason for that is because we want to be able to interact with all of this on the inside. Anything that's going to cross, if they can't interact with those fatty acid tails, they're not going to be able to get across the membrane. So obviously in this image, the ones that can cross would be like these guys, because we see them on both the outside and the inside of this cell membrane. Okay, so they must be hydrophobic, they must be uncharged, nonpolar, and they're obviously smaller than the larger orange molecules. The ones that cannot get across are large. They can be hydrophilic, charged, and polar, okay? And so again, this is where our transport proteins come into play. They can help molecules cross the membrane that couldn't cross on their own otherwise. So these guys here, too large, obviously, to cross because we don't see any here on the inside. So it's likely that either they're too large or they have a combination of the others. They may be hydrophilic and charged and polar and be too large, or maybe they're just too large in general, okay? I will add that these three terms right here, hydrophilic, charged, and polar, all should kind of start in your mind to go hand in hand. Because if something's charged, we know it's polar. And now we're just adding this additional term of it liking and interacting with water. So again, polar, charged, hydrophilic, those go together. Nonpolar, uncharged, hydrophobic, those will go together. Okay, so if anything's gonna cross, it has to have the characteristics that you're seeing there on that list: small, hydrophobic, uncharged, and nonpolar. Okay, so I think I've got a number of videos that you're gonna be looking at with this section also to help do some review. So as usual, if you have any questions, let me know. Otherwise, we will talk again in section 8.3. Bye, y'all.